It looked like I lost somebody. All right, somebody was in and now they're gone. All right, so again, we are preparing for the test tomorrow. For you all, the test is tomorrow. Hopefully you have opened up you have your worksheet downloaded. Maybe you can do split screen, I don't know. But you should have, I got too many screens going, so I'm going to be flipping back and forth. But I'm going to go ahead and get started. Let me real quick make a note of who's missing. While I'm looking at attendance, so at the meeting yesterday, they talked about how we're going to handle reviewing for for the milestone based on the scores that you had on the benchmark. So I think Monday I might have said to some of you all did very well. I think it was Troy, Kingsley, Kari, Dallas. Dallas is now just coming in, so Dallas isn't hearing me call his name. Alrighty. So I don't know whether all of you did your very best. Oh, and Christopher, sorry. Christopher, you did wonderful too. All right, Dallas, you just came in. I was just calling your name to say that you all did very well on the benchmark. When I say very well, in the A range, A range. Okay, and that's wonderful considering that we haven't taught everything on that. So that means that you should enter the exam with some confidence that you're going to do well. But how we're going to handle the review for that is there's going to be a warm-up question at the beginning of each lesson um, based on the areas of strength and weakness. And I'm going to start with the questions that on topics that we've already done. And then as we get closer to the end of March, we'll start including the topics that were on the test that we've just teaching this semester. Okay, so again, there will be a review question, or so kind of like a to-do question when you first come in. And I do mean a to-do question, so you will be expected to answer it, submit an answer kind of thing. And I'll probably put it in a Nearpod so that I can record your responses and talk about it from there. Okay. All right. So that's something to expect based on the benchmark test that you all took in December. You will take that test again just before the milestone. So just a heads up on that. And it will be the same test. So we're looking to compare what you did in December, looking to bring your scores up. And hopefully you'll give it your best shot. Neve, I'm sorry, I did not include you in that. Yours was not 90%, but it was real close. Real close. All right, so, but folks, that's 4 people who did really well. The rest out of 15. So the rest of you need to make sure that you log in every day, pay attention, and you need to be studying. You need to go back. Like I said, this is an end of course test that if you don't pass it, you have to do it again. And if you don't pass the class, you got to do the class over again and the milestone. And the milestone is 20% of your grade. So if you're just trying to get 70% in the class and you don't do well on the milestone, it's going to pull your score down to failing. Just so you know that. Okay, so you all want to try to pull your grade up to be at least 80%. That's my recommendation. Try to shoot for an 80% so that if you don't do well on the milestone, it doesn't cause you to fail. All right, not trying to scare anybody, but I'm trying to warn you that that's the way the grade for this goes. It is 20% of your grade. So sitting comfortable and say, well, in Infinite Campus, it said I was passing. That can change if you don't prepare yourself well for the milestone exam. So I think a couple of you might have just took that benchmark as a joke because we said it wasn't going to be a grade in the grade book. Okay. But it also indicated that what your weakness was. So, and some of you, it's everything. So we'll be addressing that in that fashion starting next week. Okay. All right. So just a heads up on that. All right. Worksheet. Um, so the first two, so again, we want to flip back and forth looking at the diagram. So the first two, and I'm going to do them in sets because they tend to be comparing one column with the other. So you're either getting 2 daughter cells or 4 daughter cells. So if we look at this diagram of side by side and we start at the top, the daughter cells are what you get at the end. Okay, so at the end of mitosis, you have 2 daughter cells. All right. At the end of meiosis, now this doesn't show it real good, but these, and I see these are the 4 cells that you get as a result of meiosis. This one down here is saying that if I took one, and actually they've kind of combined this picture. So if I took one cell from the female and one cell from the male and put them together, I get a zygote. So that's what this picture is showing. But what you want to understand is at the end of meiosis, you get 4 cells. And I'm going to show you, actually, let me see if I can flip over to that. No, that's that one. All right. Next, I'll show it to you later. We're going to watch, when we get to the question on crossing over, we're going to watch that. All right. Okay, so everybody got that, the first two? So you should have an X, 2 daughter cells for mitosis, an X in that column, and you should have an X in the column for meiosis for the 4 daughter cells. And the 4 daughter cells can be egg cells or they can be sperm cells. 4 sperm cells, 4 egg cells. The only difference is with the egg cells, while she makes 4, only one of them is going to get used. The other 3 will be discarded. Okay, but all 4 sperm cells will be functional. All right, the next two say the daughter cells are exact copies of the parent or the daughter cells are not identical. So flipping back again, looking at our daughter cells, looking at this is the parents up here and the parents down here for the mitosis, these look exactly alike, okay? Exact, so you got 2 cells that are exactly like the cell that they started with. With meiosis, if we're looking at these cells down here, they do not look like the one we started with, okay? We started with diploid cells for mitosis, and we started with a diploid cell for meiosis, but we ended up with haploid cells. So by the time we get here, we only have 1. And then we're also going to talk about how they're going to look a little different later on as well. So back to the worksheet. All right, so daughter cells that look exactly like the parent, mitosis. Daughter cells that are not identical to the parent, meiosis. All right, 2 cell divisions, that is meiosis. We have meiosis 1 and meiosis 2. We have 1 cell division for mitosis. Right? Now again, folks, I'm not collecting this, but hopefully you are filling this out so that you have a study sheet because these questions are on the test. You can use this to help you on the test. All right, occurs in the sex cells. Meiosis only happens in the egg cells and sperm cells. Okay, those are the gametes. All right, happens in somatic cells. All the other cells in your body are called somatic cells. Okay, so that happens in mitosis. Allows for genetic variation. That's going to be meiosis, and we're going to talk more about that in a minute because it allows you to take some chromosomes from mom and some from dad, and then switch them up. Okay, so the fact that you can choose which one of the 4 cells that you're going to fertilize. Okay, am my sound not high enough? That's going to create some. Okay, so you said this. I'm going to go back up to the top. Somebody asked me to repeat. Move that over a little bit. And hold on, let me see if this is, sometimes my volume, okay, the volume is up. Okay, maybe you need to turn your volume up. All right, so it's, okay, backing up a little bit. All right, so we have 2 daughter cells. 2 daughter cells is mitosis. 4 daughter cells is meiosis. Produces daughter cells that are exactly alike is mitosis. And produces daughter cells that are not like the parent is meiosis. Column number 2. Column number 2, meiosis has 2 cell divisions. You have meiosis 1 and meiosis 2. And let's go flashback here for a minute. All right, so this is all one event for mitosis. Meiosis has 2 events. They go up to here, produce 2 cells, and then those 2 cells divide and create 4. Okay, so there's two stages, 2 divisions for meiosis, 1 division for mitosis. Occurs in the sex cell is meiosis. That's the egg cells, and the sperm cells are the only ones that do meiosis. All the rest of the body cells are somatic cells, so that's a vocabulary word you need to know. All the other cells are somatic cells, and they do mitosis. Eye cells, hair cells, skin cells, kidney cells, all of those are somatic cells. All right, allows for genetic variation is meiosis, which means, like I said, they can change up a little bit, whereas and produces haploid cells is also meiosis. Remember, we're reducing it. We're going from 2N to 1N. So that's meiosis. Haploid cells is half the diploid. All right. Pairing of homologous chromosomes. That you're going to put an X in both columns for that. Both of them start by pairing up the homologous chromosome. That means chromosomes that are the same. And in a little while, we're going to watch a video and she's going to explain a little bit more about that. Real short, 2 short videos. And then, so an X in both of them. Our next one involves replication of chromosomes. So you're going to put an X on both of those because both of those are going to replicate at the beginning. And then after, for meiosis stage 2, they're not going to replicate. They're just going to take the 2 replications and split them. All right, this is where we're going to take a station break and look at the video for crossing over, because when I looked at the video that she had, where is she, crazy video? Hers. She skips over crossing over here. So we're going to look at a separate one. She mentions it and they cut that out. So we're going to look at it real quick over here. Okay. And then, so there's 2 back to back. So the word crossing over is what we're interested in looking at.
In meiosis, synapsis and crossing over occur between 2 chromosomes in a homologous pair, and from an evolutionary standpoint, by exchanging genetic information between the mother and father chromosome, there's an advantage of increasing variability within the gametes produced from meiotic division. Synapsis refers to the connecting of homologous chromosomes together, and this connected pair forms what we call a tetrad. Tetra meaning 4 to refer to the 4 sister chromatids present.
All right, I'm going to pause that for a minute. So we used that word homologous pairs earlier. All right, and she's going to explain this a little more, and I don't want to repeat it. But each of these, and we're going to look at chromosomes mapping with our next unit. So when they say homologous, they're the same size. They're not only the same size, but they also have the same traits on them. So different chromosomes code for different things going on in your body. So these two might be for hair and eye color. However, they look different because one of them could be coding for blonde hair, blue eyes, and the other can be coding for brunette and brown eyes. Okay, so but they're both coding for eyes and hair. So that's why they're homologous. They code for the same traits. All right. And then she's going to explain that each one of them again is a chromatid. And then she's going to explain what we mean by crossing over.
2, 3, and 4. In crossing over is the actual exchange of genetic information. So this is crossing over, where pieces of both chromosomes break off and then reattach to the opposing homologous chromosome. So the completed exchange creates recombinant chromosomes with 4 different sister chromatids, as opposed to only 2 different types of sister chromatids before the exchange. Remember, these 4 sister chromatids are then separated into 4 different gametes through meiosis 1 and meiosis 2. Interestingly, not all genes are equally likely to cross over to another chromosome. Depending on the distance between 2 genes on a chromosome, there will be a different probability of these genes separating. This makes more sense when you look at a simple diagram like this one. And so if you see crossing over between A and B is less likely to occur than crossing over between A and E, because there's only this small amount of space to cross over between A and B, whereas there's this much space to cross over between A and E. So a crossing over between A and E could happen anywhere between all this space. So it could happen here, it can happen here, it could happen here, it could happen here. There are a lot more different possibilities than between A and B. So you can see that the rule is the further apart 2 genes are, the more likely a crossover will occur and the genes will be separated.
All right, she's going to go right into the next one, I hope. I did the last time. All right, so we're going to look at, there we go. It's coming. And mine is buffering, which is why it's taking so long.
Meiosis is divided into 2 parts, meiosis 1 and meiosis 2, which are again divided into 4 more parts, prophase, metaphase, anaphase, and telophase. You should recognize that these are the same names for the mitosis phases, but they accomplish different things. Meiosis 1 is also called reduction division. Before it occurs, a cell will undergo the same process of DNA replication as in mitosis. So, when the cells begin the meiotic cell division process, there are 2 sister chromatids per chromosome. Unlike mitosis, however, the sister chromatids do not separate in meiosis 1. Instead, the homologous chromosomes separate. The homologous chromosomes are the 2 chromosomes that code for the same gene in a diploid cell. Remember, we said that humans, in each pair of homologous chromosomes, one comes from the mom and one comes from the dad. And so, for example, if we were talking about eye color, both these chromosomes will have the genes for eye color, but let's say the mom chromosome will have the genes for green eyes, whereas in the dad's chromosome, the dad will have the genes for brown eyes. So this is the difference between homologous chromosomes. One comes from the mom, one comes from the dad, but they...
All right, so I wanted to pause that for a minute to because I like this diagram. It's so much clearer than the one on Canvas. All right, so you started with the A picture, and each one of those 4 chromosomes duplicated. So we did say on our chart that both mitosis and meiosis replicate the chromosomes. So then when you look at the second picture, it looks like 2 of them, but they're connected in the middle. Okay, so it kind of looks like we've got 8 instead of 4. But 2 of them are coding for the same thing. So those 2 little ones are coding for the same thing. These 2 big ones are coding for the same thing. So what she's saying is that in this time, instead of like in mitosis, where the chromatids will break apart at the middle and separate, what's going to happen here is one of these homologous chromosomes is going to go one way and the other one's going to go the other way, so that by the time we get over here to C, we've got a big one and a little one on that one, and a big one and a little one on this one. Okay, so that's the difference, where they don't break up, the chromatids don't separate, they stay together, but the homologous pairs separate, if that makes sense. Okay, so again, these 4 that we have here, the 2 little ones and the 2 big ones, they're going to separate. So you have 1 big one, 1 little one, and that's what we mean by the homologous pairs separated, not the chromatids.
A code for different variations on a specific trait. And so when these separate, the cells go from a diploid count to a haploid count. Synapsis and crossing over also occur in meiosis 1, which is the process of exchanging genetic information between 2 chromosomes in a homologous pair. And so these 2 processes are discussed in more detail later. Once the exchange is complete, the homologous chromosomes separate by lining up randomly at the center of the cell and moving to opposite sides, and the resulting haploid cells are made genetically unique. And so in meiosis 2 divides the cells a second time to separate the sister chromatids in each chromosome. So it's very similar to mitosis in that the chromosomes line up and are separated along their centromeres. And this division maintains the haploid count in cells. However, this stage is different from mitosis in that it does not create genetically identical haploid cells. Because of the exchange of genetic information in meiosis 1, the sister chromatids in each chromosome are already different, thus 4 genetically unique.
Okay, I'm gonna pause it there because she's about to end and the picture will go away. All right, so what she's saying right here in B, pieces of the red one kind of change place with the blue one. So that when you look at this picture over here in C, you got a little tip of the blue on the red one, and there should be a little tip of the red on the other one as well. Okay, so that's what we call variation. That's crossing over. Piece crossed over from one chromosome to the other. And so when you end up with these 4 at the end, you end up with one chromosome that's red and blue. Instead of it being all red or all blue, some of them are red and blue, which means they've changed up some of the genetic information. Okay, so they're going to be different than they were when they started. So this gives you the chance. This is why you don't look exactly like mom. You might look close, mom or dad, but there's some variation because some of these chromosomes change places. Okay, all right, so that's crossing over. This is and crossing over gives you the variation in mitosis. So now imagine that these are the 4 daughter cells that we produce. These are either going to be 4 egg cells. And again, I said if it's 4 egg cells, which is the female, only one of them is going to grow up and participate in fertilization. The other 3 will disappear. If these are sperm cells, all 4 of them will grow a tail and they'll be ready for fertilization. Okay. All right. Back to our worksheet. And I think we got 5 minutes. All right, we can do this. Okay. So we were at crossing over. So we know crossing over happens in meiosis. Meiosis produces gametes. Gametes are the egg and sperm cells. No gametes in mitosis. Associated with growth and repair and asexual reproduction is mitosis. Remember, asexual reproduction, like binary fission, is simply the cell dividing up everything and splitting in two. Associated with sexual reproduction is meiosis. Chromosome number is maintained is mitosis. Chromosome number is in half. That's meiosis. Okay, that's our 2N and 1N. And both of them have spindle fibers. So you should have an X in both columns for spindle fibers are associated. You have an X in both columns for pairing of homologs, and you have an X in both columns for involves replication. Other than that, the X should be in one column or the other. Now, what they wanted you to do is take that information and put it down here, but we don't have time for that, and you don't have to do that part if you don't want to. But we are going to skip to this one. So we're looking at the difference between sexual and asexual reproduction. Sexual reproduction requires two parents. Asexual, one parent. One cell becomes two, or one plant can break off and become two plants, but that's asexual. Methods, both are methods of reproduction, so you could put an X in both columns. Creates more genetic variety is sexual reproduction. That's why we just discussed with meiosis. Asexual is going to make exactly the same cell as it started with. Short lifespan is asexual. They reproduce very quickly. Copies the DNA, both of them will copy the DNA. Crossing over, like we just discussed, only happens in sexual reproduction because it's only going to happen in meiosis. Quick reproduction is going to be asexual. I don't know if you've seen those commercials where they say one cell divides two, the two cells divide to be four, and four, but it just keeps on multiplying, but that happens very quickly. All right. More complex organisms have sexual reproduction. Simple organisms tend to be asexual. Long reproductive periods are going to be sexual reproduction. You have gestation, you have the mating process, you have everything that makes that process a little bit longer than just dividing. Autotrophic, those that feed themselves, tend to be the ones that are asexual, and heterotrophics tend to be sexual. Okay. All right. And again, you were supposed to take that
I
information.
didn't get all that.
Which one do you want me to repeat?
You were kind of
You want
going
me to
too
stop?
fast, so I only heard method of reproduction.
No, okay, starting back at the top. All right. So, one parent is asexual. Two parents is sexual. Method both. So put an X in both for methods of reproduction. Creates genetic variety is going to be sexual. So that's what we were just talking about with meiosis. Short lifespan is going to be asexual because they reproduce very quickly. Like every hour, every 24 hours, the cell is dividing, and then keep in mind that cell cycle that we talked about earlier. So there's a timing on that. So short lifespan is going to be asexual. Copies DNA is going to be both. Both of them involve DNA and genes, and you've got to make copies. Crossing over is sexual. All right, that's what we just talked about in meiosis. Quick reproduction is going to be asexual. Like I said, is the short lifespan. They reproduce almost every 24 hours. More complex organisms are sexual. Long reproduction, sexual. Autotrophs means that they feed themselves, so they are asexual, and heterotrophs tend to be sexual. Is that okay? All right. And again, I'm not requiring that you do the Venn diagram, but that's if you could take that whole list and sort it out into which ones have both and which ones are on, which ones are which, that would be that. All right, in 2 minutes, I could tell you cancer. Cancer is a mutation. Normal cells are not cancer cells. They don't grow out of control, which is the second one. So cancer is defined as cells that grow out of control. Normal cells don't do that. You can put an X on both columns for cells divide because yes, the cells are dividing, but in normal cells, they divide normally. With cancer cells, again, they are out of control, so they can keep dividing, and then that's how you get a tumor because the growth becomes localized and again becomes a tumor. It is caused by damaged DNA because somewhere along the way, the instructions are telling it to keep dividing and create these abnormal cells. So the X goes under cancer and mitosis goes under both.
You said both.
Both for cells divide and both for mitosis. Everything else is cancer. Okay. Any questions about that? And if you have any questions, other questions from your review sheets, we'll take a few minutes at the beginning of class to answer that. So write it down, but the test for you is tomorrow. Okay. All right, so I'm gonna go ahead and log off because my next class is trying to. Wednesdays feel so rushed all the time, but the next folks are moving in. Any